Okay, so let's go through a few different approaches for looking at protein diffusion and protein interactions. These are grouped under the term the F experiments. In this lecture, we're going to cover FRAP, which is fluorescence recovery after photobleaching, FLIP, which is fluorescence loss in photobleaching, FFS, which is fluorescence fluctuation spectroscopy, and then FRET that we've already mentioned. In this case, we're calling it fluorescence resonance energy transfer. So the FRAP experiment allows us to look at the mobility of molecules. So in this case, we're using the confocal laser to identify a region that we're interested in. We put the laser intensity way up for the excitation to cause damage to the fluorophores within that region. So here we've got our that's section A. We put the laser intensity way up to basically bleach all the molecules within that region. That's our bleach step here. And then we follow that over time and we look at the recovery of fluorescence back into that region. And that tends to be mobility of molecules from outside the region into there. And this is the recovery here. And we can quantify that and look at the T-half for the recovery, how much recovered, how much didn't recover, and that gives us different readouts in terms of the mobility of the molecules. Some of the applications for FRAP are things like membrane protein dynamics. So here we've got a marker which is staying within the membrane. We can bleach and follow recovery of the molecules from other regions of the membrane into here. Or things like transition of proteins across the nuclear pore complex. So in this case, we've bleached out fluorescence from within the nucleus, and then we're quantifying the return of molecules back across the nucleus through the nuclear pore, and we can quantify that. A similar technique is called fluorescence loss in photobleaching, or FLIP. In this case, we perform multiple bleaches within a certain structure. So in this case, within the cytoplasm of the cell. As we continually bleach this, we can look at compartments that are in direct contact. So if this is the cytoplasm, obviously the cytoplasm is going to be in direct contact. You can see that that is bleaching out. Whereas, say, if we look at the region of interest here on the cell membrane, we can see that that's taking much, much longer for the fluorescence to bleach out. So things we can look at are things like continuity of membranes or organelles within the cells. The next group of methods are looking at mobility of molecules. So these are highly specialized methods. And there's a number of ways we can approach these. So we have our cell sitting in there live within the confocal. And we can either park our laser beam within one spot and look at fluorescent molecules as they come in and out. So we'll see a spike as a fluorescence molecule goes through that spot, a dip as it removes out. And we can do some special mathematics here to really work out what's going on in terms of fast or slow mobility, whether things are monomeric or oligomeric. We can also do some raster scanning, which is what we're doing as we're scanning the confocal across the sample. And again, we can look at things like oligomeric state and again, mobility across the samples as well. I've mentioned FRET a few times, so we've already been through this slide, but just to remind you, we've got two fluorescent proteins, one attached to a sensory domain, one to a substrate in this example. When they're not binding, the CFP molecule will become excited and emits its fluorescence, which we can detect at this wavelength. When these bind and these two molecules come within 10 nanometers, that means that the emission energy, which would have been emitted from the CFP, is now transferred to the YFP, and that causes emission in the YFP channel. So you can see here, YFP channel emission has gone up. In order to set this up, you need to find fluorescent proteins which match or have a really large overlap between the emission of the donor and the excitation spectrum of the acceptor. So as I mentioned, CFP, YFP is a very common pair that's used for FRET experiments. In terms of the number of applications you can use for this, it's essentially endless if you can come up with a FRET pair. So as mentioned, things like protein-protein interactions, calcium sensing, where when calcium binds, you have a structural rearrangement that causes FRET. Things like kinase activity, again, when something's phosphorylated, again, that causes a structural rearrangement, which causes FRET. Or things like protease activity, where you have two fluorophores which are separated, but within a FRET distance. Once a protease cleavage site is recognized and cleaved, then that separates these two molecules out and FRET can no longer occur. So again, worth having a read into any FRET applications that might be applicable to your research. There are a number of different ways of analyzing FRET. Probably again a bit too complex to go into within this lecture series, but one of the cleaner ways of doing it is using fluorescence lifetime. If you remember, I discussed that very, very quickly in the fluorescence section of this lecture. The nice thing about this is it's independent of the wavelengths of the light, so you're looking at the fluorescence lifetime of the molecule.